Hello students, welcome back to a new video of Sunanda's tutorial. So in this video we will discuss about plasmid vector. In my previous video I have discussed about different types of vectors which are used frequently in recombinant DNA technology and biotechnology. So today specifically we will discuss about plasmid vector. So what is plasmid vector? It is small, circular, self-replicating DNA molecule. Generally, plasmid is present in bacteria as extra chromosomal sequence, and it is mostly used in genetic engineering to carry and clone foreign gene into the host cell. So using that plasmid, we can insert any foreign DNA inside the host cell. Okay. So it acts as a biological shuttle for creating recombinant DNA. Key features of this plasmid vector includes an origin of replication, from where replication can start, or called as ori site. Next is antibiotic resistant gene, which can act as selectable marker. A multiple cloning site, which allows scientists to insert, replicate and select for cells containing desired gene. So that selectable marker can be used to trace that cell is that cell contains our desired piece of gene or not. Okay. Next is key components in details. At first, ori site or origin of replication. It is a DNA sequence that initiates replication, allowing the plasmid to multiply within the host cell. Next is selectable marker, usually an antibiotic resistance gene, which helps to identify host cell that have successfully taken up plasmid. So if the cell shows antibiotic resistance, then it results or it shows that the plasmid is successfully taken up. Okay. Then multiple cloning site, a short segment with unique restriction sites where foreign DNA can be precisely inserted. Then promoter, a regulatory region that controls the gene expression inside the host cell. So these are the key components of a plasmid vector and their functions. Next, how it works. So at first, insertion, the gene of interest is caught and inserted. The desired piece is inserted into in the place of multiple cloning site of plasmid using restriction enzyme, which creates recombinant plasmid. Then transformation, that means introduction of that recombinant plasmid into the host cell, for example inside the body of Escherichia coli or E. coli. So next is selection. The host cells are grown on a medium containing antibiotic corresponding to selectable marker. Only cells with plasmid, that means having a gene of antibiotic resistance, okay, can survive. So only cells with that plasmid can survive because plasmid have antibiotic resistance gene. The surviving cells multiply, producing many copies of recombinant plasmid and potentially the protein encoded by the inserted gene. So next is the application. At first, these plasmid vector have their application in gene cloning, that means making many copies of a specific desired gene. Gene expression, it can produce protein like insulin in large quantity. Gene therapy, delivering healthy gene to human cell to treat the genetic disorder. So it is all about your plasmid vector. In my next class, I will discuss about PBR 322, PUC 19. So we will discuss about a specific plasmid vector or example of plasmid vector in details. So please stay tuned and like my videos, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.